for those of you that don't know me, my name is Ryan Martinez, and I am a PGYIV CP-only resident in the program, and I'm going to be giving my grand rounds today on some research that I've been doing in the Center for Immunology in the lab of Kristen Hogquist during residency. And I'm going to be talking about the role of Thymic B cells in health and disease. And I have no conflicts of interest. And my objectives are to summarize how Thymic B cells develop within the thymus, to identify differences between neonatal and adult thymi, and to describe the role of inflammation in the thymus. So the first thing I want to start off with is some just basic histology of the thymus and thinking about its architecture and how that relates to its function. So this is, kind of a blown up view here of a thymus lobule that you can see is separated here by this fibrovascular structure. And this individual lobule is composed of some cortex here, and as well as the medullary region. And so you can see the differences here by cellularity, and they're also defined by the unique epithelial cells that are within this region. So in the cortex, you have cortical epithelial cells, or CTECs, and in the in the medullary region, you have medullary thymic epithelial cells, or MTECs. And another thing I want to point out is some of the differences in cellularity between these, because there's slightly different processes going on that we'll be discussing later. And as well, you have these structures, these keratinized epithelial cell structures called Hassel's corpuscles that are unique to the medullary region and have some functions that I'll be discussing and hypothesizing about. The first thing I want to talk about is kind of what the thymus is doing, and the thymus really is important in generating functional T cells. The process of generating T cells is called thymocyte selection, and the goal of thymocyte selection is to generate T cells that differentiate self versus non-self antigens. And so this is really a central tenet of immunology, and this is also termed T cell tolerance. And this differentiation is important because what you're trying to do is generate T cells that have reduced reactivity to self-stimuli, so to prevent autoimmune disease, while still allowing for reaction against foreign antigens, such as those in viruses, in bacteria, parasites, etc. And thymocyte selection is a stepwise maturation process to reduce the risks of autoimmunity and to make T cells that can prevent infection. On the right here is a cartoon of what is going on during thymocyte selection. So you have a thymus lobule here, and separated into the medulla and the cortex through this cortical medullary junction. You have developing thymus cells from the bone marrow entering into the cortex originally, while the thymocytes are still very, very immature. And during this thymocyte maturation process, where they are rearranging their T cell receptor to generate functional T cells, they are starting to interact with these cortical epithelial cells, or the CTECs here. As they're maturing, they're interacting with them, and if there is no interaction with the CTECs, they're going to die by apoptosis, or if there is too strong of interaction with the CTECs, they're going to also die. As they're still functionally maturing, they finally reach a stage at which they will then transverse over to the medulla, and they will start to interact with MTECs. Again, if there's too strong of interaction, they're going to be dying, and I'll be talking about why that is. And then eventually you're going to be left with regulatory T cells or conventional T cells that will that have become a mature T cell, which will then enter out into the periphery and be able to participate in immune responses. And so that interaction process that I was talking about before is really driven by antigen-presenting cells, because that's what the thymocytes are interacting with, and that's really important in shaping T cell generation and immune repertoire. On the left here, I have just a picture of a generic antigen-presenting cell on the left, and it has different proteins in it. So that are represented by these shapes. They're able to process these proteins into peptides and present them through a MHC molecule shown here. The thymocyte, through its developing T cell receptor, is going to interact with the peptide MHC, and it's going to receive a signal. And so that interaction that I was talking about before is really this interaction between the T cell receptor and the peptide MHC molecule on the APC. This graph on the right is showing the likelihood of survival of the thymocyte when it's interacting with the TCR peptide MHC and how strong this is. So if you have a really weak interaction, you are not going to be positively selected, is what that is. And so you have a very low chance of surviving. If you have a very, very high interaction between the TCR peptide MHC, you are going to also be deleted. And so this in-between area, or this, kind of, they call it the Goldilocks hypothesis, that if you're just getting the right amount of signal, you're able to survive. This system is important for generating, again, the functional immune repertoire, because you don't want thymocytes that are able to bind too tightly to self-antigens, because these antigens are from, again, self-APC. And so these cells are going to be deleted, and so that you cannot cause autoimmune disease. Whereas these cells here are also going to be deleted because they're not able to interact with peptide MHC, and they're not able to function in the immune system. Now, there are many types of antigen-presenting cells within the thymus. They are separated anatomically, and you can see that there are some redundancies here. So you have the CTEX and MTEX, which I've already talked about, but you have dendritic cells that are defined by the SIRP alpha receptor. These are both found in the cortex of the medulla, but you can have some unique dendritic cell populations as well. There's eosinophils, mononuclear phagocytic cells, plasmacytoid DCs, and B cells, which reside predominantly in the medullary region. And so the question is, why are there so many different types of antigen-presenting cells within the thymus? And the hypothesis that we have is that, and that I think lots of people in the field have, is it's likely that each APC plays a unique role in thymocyte selection through the anatomic location and through the type of antigens expressed. And anatomically, the thymus is situated such that there are different stages of development in different anatomic locations, so that probably plays a role. And then the types of antigens, because these antigen-presenting cells are often presenting self-peptides, so ones that are being made in the cell, that the types of antigens are really important. And on the right here, I have put together how we think about types of different self-antigens. There's ones that are ubiquitously expressed, so ones that are in all cells, such as metabolic proteins, cell cycle genes, as well as tissue-specific antigens, so those that are found in the thyroid, in the pancreas, and liver. And then those also that are temporally expressed, ones that are only seen during inflammation, during malignancy, during injury. And there's some overlap between these. So you can imagine sepsis, where you would have inflammatory mediators circulating throughout the body. This will only be occurring sometimes, but it would be ubiquitously expressed. Or those during pregnancy, where they would be tissue specific in the placenta, but only, again, at certain times during life. And so there's one example that I like to bring up of why these APCs are so important, and particularly thinking about these different antigens, and that is with MTEX. And so alterations in the thymic MTEC result in massive amounts of autoimmunity. And so the MTECs express a transcription factor called AIR, and this AIR transcription factor regulates the expression of tissue-restricted antigens, so those that are found in the pancreas, the eye, lung, liver, etc. And when you lose function of AIR, it leads to an autoimmune disease called autoimmune polyendocrinopathy candidiasis ectodermal dystrophy, or APSED. And what happens is you get massive amounts of autoimmunity against these individual areas of the body that are shown here. And so these are all very unique. You can have autoimmunity against different glandular structures and different tissues all throughout your body when you lose functions in that one APC compartment. And so there has been lots of studies looking at tissue-restricted antigens and ubiquitous antigens, but there has been a lot less looking at these temporally restricted antigens. And so that is what the lab has been interested in for the past several years, is thinking about how temporally restricted antigens are generated to self-tolerance against. And so what I've been focusing on in particular is B-cell temporal antigens. And so I wanted to kind of think about this and to put this into your mind, is that the B-cell immune response actually generates these temporal neo-self-antigens, or cryptic self-antigens. And so this is simplified here on the left, but you can imagine a naive B-cell being activated and it differentiating into a germinal center B-cell or a memory B-cell or even a plasma cell. During this differentiation process, new proteins are going to be made. This is a paper looking at the transcriptional changes during B-cell immune responses, looking at mRNA levels. And between 0 and day 8.5, they have immunized the mice here to generate a germinal center B-cell response, which is shown in this gray area. And when they're looking at these particular genes that are generated in the germinal center, you can see when they're looking at different days, there's a massive increase in the mRNA levels of this. That's likely also related to protein levels. And you can see in this one protein, the gamma 1 GLT protein, it is, or transcript, is upregulated by day 3.5 and then goes away. Whereas the AICDA gene is upregulated, about peaking at day 5.5. And again, these go from not being expressed to being very, very highly expressed within the immune system and then going away. And so this is happening, all the time during immune responses. And how are we generating tolerance against these antigens? You can imagine that there's transcription factors, these enzymes, protein folding genes, and even immunoglobulins, where there are new peptides and proteins that are being formed all the time during inflammation. And why are we not generating immune responses against these brand new peptides that we've never seen before? So we decided to look into a proof of concept experiment to think about tolerance to temporal B-cell antigens to see if this was occurring or is there actually immune responses against these temporally generated B-cell antigens. So the first thing we did was we used a mouse model of temporal antigens. And the way that we did this was utilizing the CRE system. And so CRE is a genetic tool that is often bred into mice, and it is often linked such that CRE expression is driven by the expression of another gene. And so this is a common genetic tool in mouse immunology. And so what we did is we immunized different mice with this CRE peptide to generate an immune response against CRE. And we did it in these 3 different mice. The first in a wild-type mouse, the second in a mouse where CRE was expressed in all B cells. This is the gene for CD79A. And then when CRE was expressed in B cells that were undergoing class switching, so this AIDCA gene, or this is also the AID protein, which is necessary for generating class switched B cells. We then waited for 7 days and looked for CRE-specific T cell quantitation. Now, if there is tolerance against the CRE antigen, what we would see is a reduction in the number of CRE-specific T cells due to the tolerance. And when we looked at this, that is actually what we saw. So this is the quantitation of the CRE-specific T cell numbers. In the wild-type mice, there's upwards of 5,000 to 6,000 of those cells. However, when you immunize the mouse that expresses CRE in B cells or only in class-switched B cells, you have significant reduction of those cell numbers, meaning that there is tolerance against the CRE antigen in these 2 systems. One thing I want to point out is that even if it's in all B cells or only a subset of B cells where this CRE is being expressed, you still have the same number of B cells CRE-specific T cells, suggesting that tolerance is equal in these 2 cases. So we had proof of concept that there is tolerance against antigens that are only expressed in activated B cells, as in this case here. And so we had this hypothesis that B cells within the thymus were undergoing activation to generate temporal self-antigens and induce tolerance in the developing thymocytes, and that was how this system and how this tolerance was occurring. Now, it's not often thought of B cells being in the thymus, but there are a large number of activated B cells and plasma cells that are present in both the thymus from humans and mice. These are from human samples and looking at immunohistochemistry in a 3-year-old and a 29-year-old. Immunohistochemistry for CD20 shows a massive amount of B cells within the medullary region, particularly around the Hassel's corpuscles area. There's not as many within the cortical structures. As as human age, we have involution of the thymus, and it gets replaced, and the cellularity gets replaced with lipid. You can see here, but you still have a massive amount of B cells within the thymus. And so, interestingly, as you age, up to 30% of the cellularity of the thymus is actually replaced with B cells. And so these are present. And so I want to just discuss, and review B cell activation because it's important for the rest of our discussion. Overall, the goal of B cell activation is to generate high affinity antibodies with effector functionality. The way this occurs is through two mechanisms, so class switch recombination, and this generates effector function. So the class switches, to something like IgA for the gut, IgE for dealing with parasites or allergies, and then IgG for more bacterial or viral functions. So the somatic hypermutation is going to generate high affinity antibodies. So this is the insertion of nucleotides into particular areas in the immunoglobulin gene, which will then hopefully increase the affinity of the interaction between the antibody and the protein that it's going to be binding against. This occurs through interaction with B cells and T cells within lymph node or spleen. And so what happens is that a T cell gets activated and then it migrates to this area of interface between the T cell zone and B cell zone. The T cell is going to recognize antigens expressed by the B cell through TCR peptide MHC interactions, and then also deliver these signals, the CD40 ligand signals, to the B cell through CD40. Now, one thing I want to point out is at this point, the B cell is really an antigen-presenting cell, activating the TFH and receiving more signals from it. Once they have interacted, they can then migrate into a follicle and generate a secondary follicle or a germinal center. This TFH differentiates, and the B cell now starts to differentiate to start to generate the germinal center response. The class switch recombination that's occurring is also shaped by the cytokines that are within this environment, and in particular through what the TFH is making and other cells that are around in this area. The germinal center then B cell is able to mature, go into the dark zone and light zone, and undergo somatic hypermutation and generate memory B cells and plasma cells. During this entire process, the B cell is differentiating, expressing new proteins, and making neoantigens, so new antigens, as well as interacting with the T cells. So again, how during this process is tolerance being generated? So the question that I'm going to be really discussing today and try to answer are what factors are important for Thymic B cell activation, differentiation, and function, and is this any different than what we would see, say, in the lymph node or the spleen? Another question we have that we're working on is what role do Thymic B cells play in immune tolerance? So what happens when they're missing? And what happens if Thymic B cells are dysregulated? And so I'll be talking about these briefly, but really focusing on this first question. So to address these questions, we first need to identify mouse Thymic B cells. And so we digest the thymus and look at viable thymocytes. This has also been gated on single cells and live cells. We are then able to identify B cells through their staining for immunoglobulin, heavy and light chain, and excluding cells that stain for TCR beta. From this gate, we're able to then look at cells that express B220, which is a marker of B cells in mice, and then also gating on CD19, which is another marker of B cells in mice. And we have to do this because some plasmacytoid dendritic cells get through in this gate and they express B220 but not CD19. We can also identify plasma cells due to their high level of staining with immunoglobulin, but most of our analyses are looking at just the memory B cells, naive B cells, or germinal center B cells here. And so we first looked and wanted just to phenotype the B cells in the mouse and look for activated B cells. And we find that there are a large number of class switch activated B cells in the thymus. These graphs are set up such that IgM is on the y-axis, IgD on the x-axis, and this plot here, so this gate is looking at double negative cells, so B cells that have class switched. This is composed of cells predominantly of IgA, IgG2B, and IgG2C phenotype. I have looked at 3 distinct organs because I think this is an interesting comparison. In the spleen of a naive mouse, there's hardly any class switch cells, which is what you'd expect because they haven't really been exposed to a lot of antigens. In the Peyer's patches, there are germinal center B cells within these areas, and there are class switched B cells that are specific for gut luminal antigens and different bacteria that are within this area. So this is a control for activated B cells. This is really the unactivated B cell control. And interestingly, within the thymus, you have this large amount of class switch cells, actually even greater than those you see in the Peyer's patches. And so this is just a quantitation of this gate here. There have been some other work on Thymic B cells that I would just want to bring up, that this activation that we're seeing here actually occurs within the thymus. So the switch recombination products are detected within the thymus. They're not switching and then coming into the thymus. They're actually staying there. And this is also known that Thymic B cells are resident, so they don't leave once they are in there because there's no, through experiments with parabiosis, they show that the Thymic B cells, once they're there, they do not leave and exit through different lymphatics and go into the circulation. The activation process of this class switching has also been shown to be dependent on B cell antigen presentation, so that peptide MHC presentation to a T cell that is expressing the T cell receptor. It's also been important that the CD40 ligand interaction is necessary to generate these cells. And then lastly, you need this AID, which is that enzyme needed for class switching. So this is very similar to what we see out in, say, the spleen or the lymph node of just a typical B cell immune response. So there is, some similarities to what you would see in the spleen. However, at steady state, there is just a lot more activation ongoing than what you would expect to see. So next, we want to look at the germinal center reaction. And actually, what we found is that the Thymic B cells are not undergoing traditional germinal centers reactions. Again, this is the Peyer's patches, spleen, thymus. Looking on the y-axis, CD38 and GL7 is how we're defining our differentiation of B cells. This group here is composed of memory and naive B cells, and this group here is composed of germinal center B cells. And usually when cells differentiate to become germinal center, they upregulate GL7 and then downregulate CD38. And it's obvious to see here that there is this large population of this intermediate activation state within the thymus, and you're not getting any of the germinal center B cells within the thymus. And this is just quantitated here. So this is, very interesting because we do see class switching within the thymus, but we don't see germinal center formation. And so this is a little bit surprising. And so we're going to get into this in a little bit here, but we wanted to see, make sure that this process was still dependent on T cells and CD40 ligand interactions. We looked at the Thymic B cells within TCR alpha receptor knockout mice. And so these mice lack any T cells or any thymocytes. And when you look at the CD38 by GL7 population, there is significant loss here. And you lose all of your class switched B cells here. CD40 ligand signaling is also necessary for Thymic B cell class switching, as you can see from this plot where we knock out CD40 ligand on the T cell. And what's interesting is that you technically do not need CD40 ligand for this initial activation event. However, you do need it for full class switching. There does seem to be a slight reduction in the activation profile of these cells, but there's obviously something else that is driving this initial activation of the B cells. And so the CD40 ligand CD40 interactions in Thymic B cells have been studied by Luerkind's group in this paper published in Immunity in 2015. And again, they show that you need TCR and CD40 ligand to become activated and switch, and that the CD40 ligand signal is very important for B cells to become thymic APCs. So we see that as well, but this initial interaction and activation is a little bit different from what was expected. And so the takeaway points from this portion of the talk is that the presence of switch B cells in the absence of a germinal center reaction is surprising to us because that's not normally what you would see in a typical immune response. And one thing to note is that Thymic B cells actually undergo a lot of class switching, but they undergo very minimal amounts of somatic hypermutation. This paper over here from Cell Reports in 2016, I've taken some data from that, and what you can see is that within the thymus of the unswitched or switched B cells, so switched in red, the number of mutations introduced by somatic hypermutation is very low and actually very similar between the two populations. And you would normally expect these switch cells to also be undergoing somatic hypermutation, and that's not what you're seeing. However, in the mesenteric lymph node, where there are true germinal centers taking place and forming, you see that there are a lot of mutations within those B cells. And so this is very, very different. And so this is a little bit surprising. One potential hypothesis for what we're seeing is from this paper in Immunity, and they showed that class switch recombination and somatic hypermutation actually occur in series, with class switch recombination happening first, followed by somatic hypermutation. And so one hypothesis we could have is that thymic B cells are getting this initial help through the T cell interaction, but they're not getting the full T cell help they need to actually go into a germinal center, form the germinal center structure, and undergo somatic hypermutation. And so the TFHs, or the T follicular helper cells that I mentioned earlier, are really necessary for this germinal center B cell activation. On the left is a paper showing how important the TFH is. So in this mouse, they've knocked out TFHs. In this mouse, the TFHs are still present. And you can see that the germinal center B cells in this gate here are absent when TFHs are absent. And this is just showing that the TFHs are absent here and present here. You can also lose TFHs through other mechanisms. And again, the TFHs are within this circled population. You can see in ICOS knockout mice that you lose TFHs. So you'd also assume that you would lose GC B cells in the periphery, and that has been shown. And so we decided to use this ICOS knockout mouse to look to see if we would also lose our class switched B cells in the thymus, because that would suggest that they're dependent on TFHs. And what we find is that the thymic B cell activation is really independent of TFH formation. So again, we have our transitional C double positive cells in the wild type and the ICOS knockouts, and this is just quantitated here, and they're the same. And then we have the class switch cells, which are again very similar. So this is suggesting that you do not need TFHs to get this interaction. And so this is a brief model of, at this point, of what we think is going on, is that some thymocyte or T cell is delivering a T cell receptor that is recognizing B cell antigen, as well as CD40 ligand to the B cell. The B cell is being activated by the CD40 ligand interaction, but also by some other unknown factor. This activated B cell is then starting to undergo class switch recombination to generate a switch B cell, predominantly IgG2B and IgA. And then this is able to then interact with a developing thymocyte to induce clonal deletion and T cell tolerance. So the next thing we were really thinking about, is what is the thymocyte that's actually delivering this signal to the B cells? And the thymus is actually one of the first locations in the mouse where antibody switching occurs. So we were looking for, things about the thymus and trying to think about B cell activation. And this is an older paper, but what they showed in these two different mouse models, in that they compared the thymus and the spleen, and they looked at newborn mice compared to adult mice. When they looked at the different isotypes that were being made within the thymus, you can see that actually in the thymus, you have a lot of IgA, IgG3, IgG2B, and that are being made actually previously to any class switched B cells within the spleen. You can see that in the spleen, the class switch B cells greatly outnumber those in the thymus eventually, but only once you reach the adult stage. And so what is unique to this newborn thymus? And so we looked within our mice and showed that thymic B cells are actually activated within the first day of life. And so this is, confirming what that paper showed. And so with the help of a grad student in the lab, she did these kinetics, and what we found was that the transitional cells and the switch cells actually formed within that first day of life. They peaked around day 21, and then either plateaued for class switching or actually came back down at adulthood. So there seems to be a steady state of activation that occurs because the class switch cells don't just continue to increase. They reach about 20 to 25%, and then they stay there. And then the activated transitional cells also kind of stay at this 25%. And so we wanted to investigate this a little bit more, because this seemed to be a unique time during the mouse's life where you're getting a lot of B cell activation. And so the question we had is there constant activation of B cells within the thymus, or is this really happening early in life? And so what we did is we tried to address this question with proliferation studies, and we used BRDU. And we injected BRDU into an adult mouse. And BRDU is an analog of thymidine that will be incorporated during DNA replication. And we, after injection, wait 24 hours and remove the organs we were studying, looking at the B cells, and then stain for BRDU incorporation. This tests for actively proliferating cells during the last 24 hours because BRDU has been within the mouse for 24 hours. If any cell has divided within that last time and incorporated it into its DNA, we will be able to identify that. And so we find that there's actually minimal proliferation of the B cells within the adult thymus. This is a representative histogram plots looking at BRDU on the x-axis, and you can see that there is very minimal uptake within the thymus and even comparatively to the Peyer's patches. This is quantitated here on the left, and the spleen and the thymus are actually are about the same. There's no significant difference between these two, even though we know that there's a lot of activated cells within the thymus compared to the spleen, and so this was very surprising. We've also done experiments where we've given BRDU for a week just to see if there's differences in the rate of proliferation, and we still see the same results. We then wanted to look at the neonatal environment, and since BRDU can be toxic, and for the injections, we decided to look at proliferation in a different way. And we used Dappy, which stains DNA. And what we found is that Thymic B cell activation is predominantly occurring in the neonatal period. This is what the Dappy stain looks like. So you have the predominant cells that are resting here that have a certain amount of DNA in them. When a cell divides, it's going to increase the amount of DNA up to double the amount. And that is what you're seeing here is cells that are undergoing DNA replication. Here in the adult, you can see there's hardly any of those cells replicating, and this is quantitated here across multiple mice, where there's almost almost twofold increase in the amount of cells that are increasing and replicating. So that led us to questions about what is really unique about the neonatal thymus. And there's been multiple papers in immunology that have been looking at development and trying to understand how development's occurring and what changes are occurring during development. And in particular, this paper I selected from Heather Mellichar's group is that they find that the T cells generated young in life, so in particular in the neonatal period, have increased self-reactivity, and they are higher affinity for the peptide MHC. So these ones are more likely, that you could hypothesize to cause autoimmunity. And so this really occurs during the first week or so of mouse life in the neonates. And so based off of this work, what we hypothesize is that the neonatal thymus, and particularly the neonatal thymocytes, express more CD40 ligand, and that increased expression of CD40 ligand is able to induce more Thymic B cell activation. So CD40 ligand is upregulated in proportion to the amount of signal strength that they get. So again, if they are more self-reactive at this time point, we would expect them to express more CD40 ligand. And that is what we find. So this experiment here looking for the CD40 ligand expression. This on the y-axis is CD69, which is a marker of T cell and thymocyte maturation in this context. And then the CD40 ligand is on the x-axis. And this is a staining control. When we've knocked out CD40 ligand in a mouse, you can see that there's no staining. And within the neonate and the adult, we have positive staining for CD40 ligand, but you can see in the neonate that there is increased amounts of cells that express CD40 ligand. And there's also cells that express a lot more CD40 ligand, so a higher median fluorescence intensity, looking here compared to here. This is quantitated here and normalized to the adult mouse. So if the adult is 1, there's about 130% greater amounts of CD40 ligand expression, or an increase in cells by about 30% that are expressing the CD40 ligand. We have removed thymocytes that have been agonistically selected that we would expect to have high amounts of CD40 ligand from this analysis. So this is really looking at just developing thymocytes that would eventually become your conventional alpha beta T cells in the periphery. Now this just may be, something that we've identified, but within the neonatal environment, but this is not actually driving that B cell activation that we're seeing. And so to address this question, and to really ask, does the neonatal thymic environment drive B cell activation? We did direct transfers of B cells into the thymus of adult and neonatal mice. We took wild-type mice and purified their B cells from the spleen. These are adult mice, and they have a genetic label, CD45.2, so we can discriminate these cells versus the host cells. And so we either injected these straight into the thymus of an adult mouse or a neonatal mouse at around 9 days of age. We then waited for 7 days, and we looked at proliferation in this in the system. And what we found was that the neonatal thymus does drive greater B cell proliferation. This is cells that were transferred into the neonate or cells that were transferred into the adult. Again, these are the exact same cells, but just split and transferred into different mice. This has been already gated on B cells that were donated into the mouse, not the host cells. And this is gated on CD19, which is just a B cell marker, and then cell trace violet. Cell trace violet is a stain that we incubated the B cells with before we transferred. And what happens is, as the cells divide, the stain will stain the membrane. And so as the cells divide, they're going to reduce their amount of the stain by half because the membrane is going to be split. And so when a cell divides, it's going to reduce the fluorescence intensity by half, and by half again, by half again, by half again, until you get this dilution. So this is one division, 2, 3, 4, 5, 6, and then once it becomes at a low level, it's kind of hard to tell. But so there's over 6 divisions here to get to cells out in this quadrant. And so you can really see that difference here when you're trying to look at cells that have divided a lot. In the adults, predominantly, they're undivided or maybe divided once, whereas in the neonate, there's a lot of division. This is, again, normalized so that we can compare across multiple experiments. But the neonatal environment also allows for B cells to divide at about twice as many cells have divided their cell trace violet and have undergone division in the neonatal environment. We're still working on doing some of these experiments in CD40 ligand knockout mice so that we can actually say that this is really dependent on the CD40 ligand. But we're trying to close that loop and to try to really tie up that mechanism. And so I want to bring together again, just to kind of put everyone back into context of what we're discussing. And so we have this neonatal thymic environment where you have this highly self-reactive CD4 single positive cells that are being generated. These cells are upregulating large amounts of CD40 ligand. They're interacting with their B cells. We don't know what the fate of these thymocytes are at this point. We don't know if they're undergoing negative selection or if they're surviving or really what's going on. So that's something in the future that we're interested in looking at. And we know then that these B cells, once they've received that signal in this neonatal state, they become activated. We still know, that there's something that there's another step here that we're missing, but they become activated, they undergo class switching, and then they're able to interact with other developing thymocytes that are recognizing these temporal antigens. The other thing we're trying to think about is this class switch recombination. So cytokines are necessary for shaping and directing which class isotype you go down. And so what cytokines are being expressed and how are they being expressed? And so the next part of the talk, the last part of the talk, I'm going to be thinking about more this, is what is that other signal that B cells need for activation? And so the way we address this is through an RNA-seq approach. So we compared the class switch cells in the Peyer's patches and the thymus. And why we did this is because we thought that these would be the most activated cells. And if the thymus has a unique activation pathway, that we would be able to see it when we compared a traditional germinal center pathway versus this untraditional activation pathway. And this project and this part of the project was actually performed with LMP resident research funds that I was awarded during my first year. So I'd like to thank the department for that. And so when we looked at pathway analysis, we find that of this RNA-seq, we find that the class-switched Thymic B cells are enriched for pathways upregulated during inflammation. How you read this here is we've organized the pathways into normalized enrichment scores. So if this pathway is upregulated either in the thymus to the left or the Peyer's patch to the right. and I've also color-coded it. And these are all significantly upregulated with a p-value less than 0.05. And I just want to draw your attention to these top targets are actually all inflammatory targets. So you have interferon alpha, interferon gamma, interferon beta, just general inflammatory responses, and the toll-like receptor cascades. And so this is very surprising because the thymus, I always thought of it kind of as this more or less sterile environment where T cells are, undergoing maturation. It's kind of this nice, healthy nursing environment for these developing cells. But it turns out there's actually a lot of inflammation. And so we focused on interferon within the thymus, and there's a couple of reasons why we did that. And so first I want to talk about interferon. So interferon is a set of proteins that can be separated into 3 distinct types, type 1, type 2, and type 3. They are separated by these types depending on which receptor they interact with and which heterodimer receptor. So there's the IFNAR for type 1, the interferon gamma receptor for type 2, and then the interferon lambda receptor for type 3. And these are termed interferon, these proteins, because they interfere with viral replication. They're secreted by cells when they've been infected with virus, and they help to shut down viral proliferation and survival. And so interferon really initiates a defense response to virus. The way this does this is through it's by regulating transcription of interferon-stimulated genes, which will then protect the cell. And so, example of this is MX1. This is a gene that's important for this interferon process. And the reason why we were started to think about the interferon within the thymus is from this paper in 2009, where they had an interferon beta luciferase reporter. And so they were looking at interferon beta production throughout the mouse. You could inject substrate for for luciferase into the mouse and then do whole mouse imaging. And when there was this red color, that would be where interferon beta was being produced. And interestingly, when they looked at the steady state, you can see that the majority or the highest amount of interferon was actually being produced in the thymus. And again, this is interferon beta, but there's a high amount of it going on in the thymus just under normal conditions. And this is the control mouse. So based on this, we were interested to see if the Thymic B cells were sensing interferon. And the way we did that was with a reporter mouse, and we found that they are expressing interferon-stimulated genes. We had a wild-type mouse, or a mouse that was a GFP knock-in mouse. So when MX1 was expressed, it would express GFP. And again, MX1 is an interferon-stimulated gene. These are an overlay of the flow cytometry plots from the Thymic B cells, and looking at GL7. So above here, these cells are those activated B cells within the thymus. And when you look at the GFP signals, so this is when the cells that are sensing the B cells that have been exposed to interferon, you can see that all of these GL7 positive cells here have seen interferon. And there's actually a large portion of cells that have not become activated by GL7 marker, but also express interferon. And so this was good to see, but it also shows that interferon may be potentially helping activation. And this is in collaboration with Oscar Salgado Barrero and Thera Lee that were also working on this interferon project for a different component within the thymus. And so this led us to the question, by seeing these data, is that how does interferon impact thymic antigen-presenting cells? And inflammatory temporal antigens are generated by interferon signaling and are expressed in the thymus. So this potentially could be a way that temporal self-antigens are being regulated. So we decided to look big into trying to look at the whole thymic environment. And so what we did was we did cell sorting of MHC class 2 expressing cells from the thymus, followed by a single cell RNA-seq to look at the transcriptional profile. And to try to understand how interferon was impacting the thymic APC, we combined these cells from either wild-type mice, IFNAR knockout, or IFNAR knockout interferon lambda receptor double knockout thymi. So these ones would be deficient in type 1 signaling. These ones would be deficient in type 1 and type 3 interferon signaling. Now, B cells are a large component of the class 2 expressing cells. So we were able to look at global, but also I could analyze the B cells. And so the hypothesis we had was that interferon signaling would alter the thymic B cell activation and function. And so what we find is that there are transcriptionally distinct groups of thymic APCs. This here is a UMAP plot, and so the individual dots represent individual cells, and these are graphed based on how similar they are in the genes that they express. And so you get these clusters of cells of interest. And so to me, these ones were the ones I was most interested in, the B cells, but as you can see, there are lots of other APCs that we've previously mentioned. And so I wanted just to look first at the B cells. And what we found is that type 3 interferon is necessary for Thymic B cell phenotype. And the way we found this was that I first looked at the B cells and I split them up and tried to cluster them into distinct groups. And so we made this cluster 0 and cluster 1. Again, this is only B cells with each dot representing a single B cell. Then when I separate them back out into the original genotypes, what we can see is that the purple cells, which is from the wild type mouse, are scattered between cluster 0 and cluster 1. In the green, you can also see that they are scattered between the two clusters, but the orange, which are the double receptor knockouts, they're really only in cluster 0. And so it seems that the cluster 1 are either need the interferon lambda receptor, or the cells have left the thymus and they're not able to take on this phenotype when they don't have this interferon lambda receptor expression. When we tried to identify, what the genes were that were different between these clusters, what we find is that the type 3 interferon signaling is associated with Thymic B cell class switch recombination. So again, cluster 0, cluster 1, these are now broken up by the individual genotypes, and you can see that a lot of the IgA, so when it's blue, that means there's a lot of IgA expressed. And so in the wild types, that's the cluster 1, so there's activation state. You can see this in the IFNAR knockout, looks almost identical, but really these cells are really lacking in these double knockout. And so this is also recapitulated when we look within these mice for IgA switching, you almost have no IgA switching. So thinking about a model now is that everything is very similar. We have now filled this in a little bit more with some markers that we've identified on the activated B cells that are unique from the RNA-seq, but we've also thought about interferon lambda receptor, which we think is important and potentially is expressed on the B cell, though we're still investigating this. And we know that MTECs are making interferon lambda. There's just recently been a paper from this. And so we think what's going on is that this thymic environment is actually very inflammatory. It's making interferon beta, it's making interferon lambda. This interferon then is helping to activate the B cells and is helping this process along. And so we're still trying to investigate how interferon lambda is impacting B cell function, and also trying to understand what happens when you lose tolerance to B cells. So that's the end of the data portion. I just wanted to end on one other last thing. And this last question here is, what is occurring when thymic B cells are dysregulated? And so this kind of leads to clinical utility and interest in the future for us to try to think about how this is impacting human health. And we have turned to myasthenia gravis and the thymic B cells that are present there during myasthenia gravis. And so myasthenia gravis is an autoimmune disease that's mediated by anti-acetylcholine receptor antibodies. And the anti-acetylcholine receptor B cells are often found in the thymus. And what's interesting is that thymectomy is often a treatment for myasthenia gravis and does help a large subset of patients. And when they look at thymectomized patients with myasthenia gravis, they see that there's follicular hyperplasia in about 70% of younger patients with myasthenia gravis. There's different subsets within with myasthenia gravis, but this is found in a lot of patients. On the left, you can see some of the histology from these samples. You can see germinal center B cells forming within the medullary region. They're stained for CD20, and you can see, again, this is a B cell cluster here with follicle formations, and they're even staining for CD23, showing that they have a nice FDC meshwork. So these are true germinal center B cell responses that we typically don't normally see in a healthy thymus, but are being seen in the case of myasthenia gravis. And so I wanted to bring this together because I think that actually we have, from our system, we actually have some interesting data from humans that shows that interferon pathways may be dysregulated in patients with myasthenia gravis with follicular hyperplasia. This is a paper where they looked at array data from the thymus of patients with and without myasthenia gravis, and they see that there is a large group of genes that are interferon-induced that are upregulated that are not seen in healthy people. And then this same group several years later showed actually that when you treat medullary thymic epithelial cells with different types of interferon, you actually upregulate the acetylcholine receptor on the MTEX. And so they thought, this is hypothesized that this is actually the source of antigen for initiating the immune response against the acetylcholine receptor, so that B cells are then starting to be able to activate this, activate and respond to this antigen. And so it may be a combination of interferon that is helping the B cells to class switch and be activated, and along with that activation, if there's now more of this acetylcholine protein being made, potentially that is a route of pathogenesis for this disease. And so future studies are going to, that I'm interested in performing, are going to determine if exogenous interferon alters Thymic B cell activation and germinal center formation, and if this leads to, those more typical germinal center structures and formation of antibodies and autoreactive antibodies in particular. And so with that, I'd like to stop and acknowledge the Janequist lab, particularly Elise Breed, for really starting off this Thymic B cell project and helping me with some of the data. I'd like to thank Oscar Salgado Barrera and Thera Lee for a lot of work on this interferon project that they are also investigating. I'd like to thank the LP residency program, Dr. Dolan, Dr. Morose, Dr. Linden, Dr. Nelson, Dr. Fieri, Dr. Furcht for their support over the last several years and mentorship. And then Christy Hensler in the MSI for the help with the RNA-seq and trying to run the pipelines, and me emailing her and she getting back to me very, very quickly. UMGC for doing a lot of the sequencing, and then of course Chris for being a great mentor, and then also Steve and Sarah for all their help in lab meetings and discussions that we have. And so with that, I will take any questions.
Well, Ryan, I'll start out. I'm John Crossan, as you know. Fascinating talk and really great work. Thank you very much. The question is more of a clinical question. As you know, transplant clinicians are very anxious to develop a satisfactory tolerance model for the specific allogenic organ that they're going to transplant. How does your work influence the concept of developing tolerance under the setting of transplantation?
Yeah, I think that's a good question. I mean, the tolerance you need, unfortunately, in that we're seeing, we think we need, is you kind of need a shadow of that, whatever you're going to be. I guess it's like the shadow of what you're going to be seeing, but you need it in the thymus so that you can undergo the correct thymic selection mechanisms. So, I mean, I'm not really sure, because all of it is kind of a temporal antigen, because all of a sudden it wasn't there, and now all of a sudden it is there. So I'm not sure. I mean, I think in the mouse model, I mean, sure, you could inject things into the thymus. You could have B cells that express the right HLA molecules or class 2 molecules and activate them within thymus, and that should be able to induce tolerance. But as far as clinically, it's still a real challenge. I mean, I think Tregs and trying to educate regulatory T cells on HLA antigens may be a way to go, but I'm not really sure at this point.
Ryan, this is Chris Pinnell. I have two questions. One is, what maintains the residency of the B cells in the thymus? And then the second is, are the antibodies that they secrete functionally important?
Yeah. We are not sure as to how the B cells stay resident. We were hoping with the RNA-seq that we would identify some chemokine or some group of chemokines that they may use. Nothing really jumped out at us. There's a couple of markers. They express high amounts of CCL22, CXCR3, and yeah, I think those are really the main ones that seem to be differentially regulated compared to those out in the periphery. So we're not really sure. We know that there is a niche environment though for them. So if you transfer B cells, into a B cell deficient mouse, that you get more of them into the thymus. There seems to be some regulation and some factor. There's been some reports that BAF may be important, but I'm not sure exactly. That's maintaining the number there, but I'm not really exactly sure how they're retained there. As far as, sorry, can you remind me of the second question again?
Yeah, are the antibodies that they secrete playing any functional role, like in, mediating phagocytosis of antigens, or?
Yeah, so so there has been some reports in humans that actually after immunization, so after flu, after measles immunizations, you actually have a subset of plasma cells that survive in the thymus and survive for long times and are secreting antibodies. So there seems to be some role for just general antipathogen responses. Other people have shown that there is an enrichment for self-reactive B cells within the thymus, so those that are making more double-stranded DNA antibodies. So it's kind of a mix. So it seems to be that they're potentially important for preventing future pathogenesis from viruses and bacteria, but also there seems to be a reservoir for potentially self-reactive, autoreactive B cells as well. And so we haven't really discriminated yet if they are for good in protection or bad for generating autoimmune disease.
Are B1 cells enriched in the thymus?
No, they're not.
Okay, thank you.
Mm-hmm.
Hi, Ryan. This is Dr. Adey here. I really enjoyed your presentation. Very interesting. Thank you. My question has to do with CD38. My understanding is that this is expressed usually in post-germinal center B cells, but it looks like you used it to get in on naive B cells in your series. So is it true that they are really expressed in naive B cells, and is that true in mice only, or carry over to humans?
Yeah, so unfortunately, I think it's a difference between mouse and humans. So in mice, it is expressed on naive and memory cells. There's not, and whereas in humans, yes, it's expressed in a different fashion and regulated in different ways. So it's a difference between the two models, or between a mouse model and the real life human.
Okay. And then the second question is on the definition of neo self-antigens.
Mm-hmm.
So does that cover everything that the activated B cell produces? So you mean all the cytokines and immunoglobulins, or is there a specific definition that makes this antigenic to begin with and then qualify to be neo self-antigen? Yeah. And if it covers all the productions, including cytokines, then will it be fair to extend the definition of neo self-antigens to other cells outside of B cells that also produce other immune molecules?
Yeah, so I think, neo self-antigens or cryptic self-antigens, I think are I'm using it in a very general term. I'm just saying anything that the B cell previously did not produce but is producing during activation or differentiation. And so, yeah, you can imagine that a B cell will express new cytokines that it did not during the resting state. But that is not to say that other cells within the body or other immune cells will also not express that. So things such as, or like interferon, can be expressed by a lot of cells. And it may be a neo self-antigen for multiple types of cells. So I think maybe thinking about, that there's going to be overlapping neo self-antigens between different immune cell subsets. That some may be unique. So germinal center transcription factors are probably going to be very unique to the B cell, whereas if production of cytokines are going to be more shared across different inflammatory cell types.
Thank you.
Okay, we are going to cut it off. We have the quality meeting, which starts at 9. So I am going to close out of this. Thank you, Dr. Martinez. Great job. So we will see you all at 9. Thank you.